Think of our bodies as intricate networks of pathways and signals. These networks are constantly buzzing with activity, keeping us alive and functioning. Now imagine drugs as messengers carrying specific instructions to different parts of this network. But for these messages to be delivered correctly, the messenger needs to have the right shape. This is where the fascinating world of drug design comes into play. In the realm of medicine, shape is everything. Just as a key fits into a specific lock, drug molecules must precisely fit their biological targets. These targets are often proteins or enzymes that play crucial roles in our bodies. If a drug molecule doesn't have the right shape, it won't be able to interact with its target, rendering it ineffective. The development of new drugs is a complex and meticulous process. Scientists spend years studying the intricate shapes of biological targets and designing molecules that can interact with them effectively. This involves understanding the chemical properties of both the drug and the target, as well as the forces that govern their interactions. The shape of a drug molecule determines its ability to bind to its target, influencing its potency, selectivity, and overall effectiveness. In essence, the shape of a drug dictates its destiny, determining whether it will be a successful treatment or a pharmaceutical flop. Imagine a lock and key. The key's intricate grooves and teeth are designed to match the lock's internal mechanisms perfectly. Only when the key aligns flawlessly with the lock can it turn and grant access. This elegant analogy perfectly illustrates how drugs interact with their targets in our bodies. Drug molecules are like keys, each with a unique shape designed to fit a specific lock, which represents the biological target. These targets can be receptors on cell surfaces, enzymes involved in metabolic processes, or even DNA itself. The interaction between a drug and its target is highly specific, much like the unique fit between a key and its corresponding lock. When a drug molecule encounters its target, it forms chemical bonds that hold it in place, like the teeth of a key engaging with the tumblers of a lock. This binding event triggers a cascade of molecular events, ultimately leading to the drug's therapeutic effect. The precision of this interaction is crucial. If a drug binds too weakly, it may not elicit the desired response. Conversely, if it binds too tightly, it can cause unwanted side effects. The key lock analogy highlights the importance of shape complementarity in drug design, emphasizing the need for molecules that fit their targets with both precision and affinity. Now, let's delve into a fascinating aspect of drug shape known as chirality. Imagine your hands for a moment. They are mirror images of each other, yet you can't superimpose one directly onto the other. This unique property of handedness is called chirality. In the world of molecules, chirality plays a crucial role in determining a drug's activity. Many drug molecules exist as chiral pairs, meaning they come in two forms that are mirror images of each other, just like our hands. These mirror image molecules, called enantiomers, may seem identical at first glance, but their subtle differences in spatial arrangement can have profound effects on their biological activity. Think of it like this: trying to fit a right-handed glove on your left hand. It just won't work. Similarly, one enantiomer of a drug may fit perfectly into its target, while its mirror image may not bind at all, or worse, bind to the wrong target, leading to unintended consequences. Chirality is especially important in drug development because our bodies are inherently chiral environments. Our proteins, enzymes, and receptors are all chiral, meaning they can distinguish between different enantiomers of a drug. This selectivity arises from the fact that biological systems are themselves made up of chiral building blocks, such as amino acids and sugars. To truly grasp the impact of chirality, let's look at some real-world examples. Carvedilol, a drug used to treat high blood pressure and heart failure, exists as a mixture of two enantiomers. One enantiomer effectively blocks certain receptors in the body, contributing to its blood pressure-lowering effects, while the other enantiomer is much less active. Another striking example is ketamine, an anesthetic and increasingly popular treatment for depression. Ketamine also exists as two enantiomers, and they have drastically different effects. One enantiomer, known as S-ketamine, is a more potent anesthetic and has a better safety profile, while the other enantiomer, R-ketamine, is associated with more side effects and psychoactive effects. These examples highlight the crucial role of chirality in drug action. By separating and studying the individual enantiomers of chiral drugs, scientists can gain a deeper understanding of their therapeutic benefits and potential risks. This knowledge paves the way for the development of safer and more effective medications that target specific pathways in the body with greater precision. Section 5. The enantiomer advantage, potency and duration of action. The distinct three-dimensional shapes of enantiomers mean they can interact with our bodies in vastly different ways. This difference in interaction can manifest in several ways, most notably in a drug's potency and how long its effects last. Let's break this down. Potency refers to the amount of a drug needed to produce a specific effect. One enantiomer might be much more potent than its mirror image, meaning a smaller dose is needed to achieve the desired therapeutic outcome. This difference in potency can be significant, sometimes differing by tenfold or even more between enantiomers. Duration of action, on the other hand, refers to how long a drug's effects last in the body. Again, enantiomers can vary widely in this regard. One enantiomer might be metabolized and eliminated from the body quickly, while its mirror image could linger for an extended period. This variation in duration of action can have implications for dosing schedules and the potential for side effects. Understanding these subtle yet crucial differences between enantiomers is paramount in drug development. By optimizing a drug's enantiomeric composition, scientists can enhance its therapeutic benefits while minimizing unwanted side effects. This knowledge is driving a trend toward developing drugs that contain only the most active or beneficial enantiomer, leading to safer and more effective treatments. Section 6. Racemic mixtures, a double-edged sword in medicine. For much of pharmaceutical history, drugs with chiral centers were often produced and administered as racemic mixtures. These mixtures contain equal amounts of both enantiomers, and while this approach may seem practical, it can be a double-edged sword. On the one hand, using racemic mixtures can be less expensive and less time-consuming from a manufacturing standpoint. It's often simpler to synthesize and purify a mixture of enantiomers than to separate and isolate the desired enantiomer. However, this convenience comes at a potential cost. The problem with racemic mixtures is that the inactive enantiomer isn't always truly. Section 7. The promise of enantiopure drugs, a future of precision. The limitations of racemic mixtures have spurred a growing movement in the pharmaceutical industry towards developing enantiopure drugs. These drugs contain only the single desired enantiomer, offering several potential advantages over their racemic counterparts. Firstly, enantiopure drugs offer enhanced selectivity, meaning they're more likely to interact with their intended targets and less likely to bind to other targets, reducing the risk of off-target effects and potential side effects. This enhanced selectivity is particularly important for drugs that target the central nervous system, where even subtle off-target interactions can have significant consequences. Secondly, enantiopure drugs often exhibit improved efficacy compared to racemic mixtures. By eliminating the inactive or less active enantiomer, the desired enantiomer can exert its therapeutic effects more efficiently, potentially leading to better treatment outcomes. Finally, enantiopure drugs can simplify dosing regimens and reduce the likelihood of drug interactions. With only one active enantiomer, the drug's pharmacokinetic profile is more predictable, making it easier for doctors to determine the appropriate dosage and minimize the risk of adverse events. Section 8. Challenges and opportunities in chiral drug development. While the potential benefits of enantiopure drugs are undeniable, their development and production present unique challenges. Separating enantiomers can be technically demanding and costly, requiring sophisticated techniques such as chiral chromatography or asymmetric synthesis. Moreover, regulatory agencies are increasingly demanding more stringent testing and evaluation of individual enantiomers, even for drugs that have been on the market for years as racemic mixtures. This increased scrutiny, while necessary to ensure patient safety, adds another layer of complexity and cost to the drug development process. Despite these challenges, the field of chiral drug development is brimming with opportunities. Advances in synthetic chemistry and separation technologies are making it easier and more cost-effective selective to produce enantiomeric drugs. Moreover, growing awareness of the importance of chirality among researchers, clinicians, and regulatory bodies is driving the development of more selective and effective medications. Section 9. Conclusion: Embracing chirality for better medicines. The shape of a drug molecule is not merely a matter of geometry, it's a matter of life and death, health and disease. Just as a key unlocks a specific door, the right drug molecule, with its precise shape, can unlock the body's healing potential. Understanding chirality, this fascinating aspect of molecular shape, is crucial for developing safer and more effective medications. By harnessing the power of chirality, we can move away from the limitations of racemic mixtures and embrace a future of enantiomeric drugs, ushering in a new era of precision medicine tailored to the unique needs of each patient. As we continue to unravel the intricate dance between drug shape and biological activity, we unlock the potential for groundbreaking treatments that transform human health.